In this video, I'm going to talk about the electrophysiology of neurons. The electrophysiology of neurons happens in three major steps: resting potential, local potential, and action potential. And this is then followed up by a process known as synaptic activity or synaptic transmission. So this picture just kind of is giving you a visual for those three steps plus the synaptic activity that I mentioned. So resting potential is something that occurs all along the neuron. Local potentials tend to happen at the dendrites or soma of the neuron, whereas an action potential occurs along the axon of a neuron. And synaptic activity happens at a synapse, which is where a neuron and another cell communicate. So this next little animation just gives you an overall idea of what's happening during the electrophysiology of neurons, and then what happens during synaptic transmission or synaptic activity. So we start off at resting membrane potential, which is negative 70 millivolts, and then let's see what happens when we change that. Alright, so we start off at resting membrane potential. Something comes in, some sort of stimulus changes the resting potential from negative 70 to negative 65 to plus 35. Right, so we've gone through a series of voltage changes. And the voltage changes at the very end cause these little Pac-Men to be released by the ends of the neuron. The little Pac-Men are representing neurotransmitters. So neural activity is showing us how a neuron communicates with another cell. Going back to this picture here, right? This neuron is communicating with another cell over here. The other cell is going to be a neuron in our example, in the examples that we run through, I should say. But the other cell could also be a muscle cell or a gland cell. Neural activity uses both electrical and chemical activities. A lot of people tend to think that neural activity is all electrical in nature. I mean, there is an electrical component to it, but there's also a chemical component too. The electrical activities are changes in the transmembrane potential, changes in the voltage across the cell membrane. The chemical component is the release of the neurotransmitters at the very ends of the axon. So a quick review of transmembrane potentials. Neural activity does involve changes in transmembrane potentials. Don't forget, a transmembrane potential is just a charge or a voltage difference between the inside and the outside of a cell. Resting membrane potential is the transmembrane potential of a cell at rest. Cells are said to be polarized at rest. They're polar like a battery, polar like the earth, meaning there's a positive side and a negative side. For cells, the positive side is on the outside of the cell and the negative side is on the inside of the cell. The average resting membrane potential for a neuron is negative 70 millivolts, and the term negative is used because we always talk about the inside of the cell as compared to the outside of the cell. So the inside is negative as compared to the out, and so we use the term negative 70 millivolts to talk about resting membrane potential. Resting potential is created in part by the sodium-potassium exchange pump. Transmembrane potentials can change, or else they'd be really boring. They change when ion channels open, and then the ions follow their concentration gradient, going either into or out of the cell, and that changes the voltage. Changes in transmembrane potential occur in response to a stimulus of some kind. The stimulus is usually either a chemical stimulus, a mechanical stimulus, or a voltage stimulus. These stimuli open gated ion channels, and opening a gated ion channel can cause one of two things. It can either cause a depolarization or a hyperpolarization. A depolarization is becoming less positive, right? It's becoming more positive, whereas a hyperpolarization is becoming less positive. This series of pictures just kind of nicely illustrates the different types of gated ion channels that are out there. So our different types of gated ion channels are chemical gates, also known as ligand gates, and then we have voltage gates, and then thirdly we have mechanically gated channels. Mechanically gated channels open due to mechanics, which would be like some sort of physical movement. Take a minute and look at these panels, A, B, and C, and see if you can figure out which is the chemically gated ion channel, which is the voltage-gated ion channel, and which is the mechanically gated ion channel. In panel A, we see a chemical, acetylcholine in this case, coming in and binding to the channel. And once it's bound, it opens the channel. So this would be a chemically gated channel. I think B is pretty much the easiest. You see a change in voltage that causes the opening of the channel. And here in C, we have some sort of applied pressure, a physical movement, opening the channel. That's a mechanically gated ion channel. All right, so now we're going to take a look at the electrophysiology of neurons. So the first step is the resting potential. The resting potential is the transmembrane potential of a cell at rest. The second part to the electrophysiology of neurons is the local potential. A local potential is a change in the transmembrane potential, and this is usually experienced at the dendrites or the cell body of the neuron. It's called a local change because it doesn't travel very far. It stays localized to where the stimulus is applied. What causes a local potential is a stimulus, and it's usually a stimulus that opens chemically gated or mechanically gated ion channels. Voltage gated channels usually are not involved with local potentials. Voltage gated channels are important for the next step, action potentials. An action potential in a neuron is also known as a nerve impulse or an electrical impulse. And this is a moving, dramatic change in voltage along the axon. And this moving change goes all the way to the ends of the axon. This particular step requires voltage-gated ion channels. And what starts an action potential is the previous step, the local potentials. Action potentials and local potentials are both changes in the transmembrane potential, but action potentials are much more dramatic. You get larger changes in voltage, and action potentials move. Local potentials are local. The electrophysiology of a neuron is followed by synaptic transmission, also known as synaptic activity. Once the action potential reaches the end of an axon, it causes the release of a neurotransmitter. The neurotransmitter then travels across the cleft where it binds to another cell and causes something to change in the other cell. If the cell on the receiving end, called postsynaptic cell, is a neuron, then the binding of the neurotransmitter to the postsynaptic neuron will cause a local potential in the postsynaptic neuron.